非常感谢刘博宇主任的精彩分享。那我相信在这个前4场的这个 talk 中间的话，大家也已经累积了很多想要跟这个演讲者交流的一些问题。那接下来呢，就是我们 panel discussion 的时间。那在这个 panel discussion 的话，我们很荣幸邀请到台大医院感染科的这个申万辉教授来为我们做这个panel discussion 的主持。让我们掌声欢迎申万辉教授。
Okay, thank you for your introduction. I'm Dr. Wang Hui from National Taiwan University Hospital. It's my honor and pleasure to host the panel discussion. I think mNGS is the next sequencing of typing of unknown pathogen or emergent infectious disease or some of difficult to diagnose pathogen in Taiwan. I think the technique will be broad, widely used in Taiwan hospital. Before we have the panel discussion, I would like to invite the attending in the auditorium to have some comment and question for our foreign speaker or for our Taiwanese speaker. Is there any question or query coming from the audience? Professor Chu?
I'm Dr. Nanchang Chiu, also Dr. Chiu. And I'm a pediatrics infectious physician and also a pediatric neurologist. So my main interest will be the encephalitis. And as we know that encephalitis, not only the pathogen, but also autoimmune encephalitis become a very important part for the diagnosis of the reason why this kids get the encephalopathy. And as you have already mentioned that this new technique, not only for the pathogen, but also have some development for the detection about the autoimmune response. Can you give us more comment about that? Thank you.
Sure, I would be happy to. So, you know, I should also remark is that this has been an interest of my collaborator, you know, Dr. Michael Wilson as well. So perhaps he can also chime in afterwards. But the general idea is that with clinical metagenomics, we run both DNA and RNA libraries. And so when we run RNA libraries, for metagenomics, we typically run them so that we can detect RNA viruses. Because if you do only DNA metagenomics, you don't really have the capacity of identifying RNA viruses, which are a very large component of certain types of infections, such as neurological infections and perhaps respiratory infections as well. And because we do metagenomics, when you look at the RNA libraries, effectively, when we're doing an RNA-seq experiment, we're doing RNA sequencing. So the idea behind that is we can, instead of looking at the microbial reads, the reads corresponding to potential pathogens, we're looking at human reads. And we're looking at essentially the what we call the transcriptome profile or the RNA host response of the patient. And so the idea is that we can leverage that by analyzing the RNA-seq data, RNA sequencing data. We can develop predictive models using machine learning by which we can, if we have enough examples of each of the different infections and non-infectious conditions like autoimmune disease, in principle, the idea is that you can use the host response profile to be able to distinguish between, for instance, infectious causes and non-infectious causes, or perhaps to distinguish between bacterial and viral infection, or other types of comparisons. So ultimately, this is a very powerful approach because we're not doing another test. You don't have to do any more work. If you're already doing RNA metagenomics for RNA viral detection, we're simply leveraging the data that's already present. The data has already been collected. But maybe I'll pass it over to Dr. Wilson to comment.
No, I think that summarizes it really well. I think the, you know, I would just add, you know, there's also there's the benefit of just the negative sequencing result for, an infection. That gets you part of the way there, along with the clinical context to be able to say that it's likely not infectious. And I think, there are there's a number of studies out there already with doing RNA sequencing, especially of blood, to be able to distinguish different disease types like infection versus not, or you can find papers on, latent versus active TB based on the host transcriptomic signature in the blood. And I think we've been hopeful that, you could generate similar types of data sets from spinal fluid. It's taken longer, I think, because it's taken longer to get good quality RNA sequencing from spinal fluid because the input amount of RNA is so little, and also just because many of us who do metagenomic sequencing tend to skew our patient populations towards highly unusual cases, and so I think it's taken another effort to collect more larger groups of patients with known conditions. So enough patients with known causes of autoimmune encephalitis, known TB meningitis, cryptococcal meningitis, et cetera. So just getting the patient cohorts together is taking some work, but fortunately, just like has been seen in blood, there seems to be a real way forward with CSF as well.
Okay, thank you. Professor Chen.
Thank you for all for our distinguished speaker here. On behalf as an ID physician and the infection control personnel, I thank you for give a hope, a landscape for combating this challenge. I have a question regarding the imbalanced data analysis. As Professor Zhu and Wilson, you mentioned that the trace element of the potential pathogen in limited volume of clinical sample. However, it turned out to be the causing pathogen. Among numerous, might be human DNA or others. So how to handling from sample processing as well as data processing? Because for many years, when developed such kind of platform for more sensitive to detect this kind of pathogen. This is two-level question. One is for wet lab sample processing. The other is data processing for all our four speakers.
Yes, I think it's a very important question about the achievement of the mNGS result. So is there any comment from all the speakers?
I could start and just on the sample processing, with the RNA sequencing libraries, we've done some work to deplete mitochondrial RNA sequences, which can, depending on the library, can form a large proportion of the total number of reads that come out. And in early days, there was a talented, again, Wei Gu and Emily Crawford developed the technique using CRISPR to deplete cDNA molecules in the sequencing library that align to human mitochondrial sequences. And by removing those cDNA molecules, that allowed for an increase in the proportion of pathogen reads while still preserving kind of the overall human transcriptomic data. And there are now, QIAGEN makes similar depletion. Those depletion tools are now commercially available that really effectively will knock out those sequences from the libraries, and that either allows you to sequence with less depth and or sequence at the same depth, but have a higher sensitivity for pathogen detection. And I'll let Charles talk about the analysis front.
Yeah, I think there are, as Dr. Wilson described, there are different ways that you can enrich or deplete for enriching or for your target. For the target gene or the target pathogen that you're looking for. For RNA libraries, what we routinely do is we pre-treat, we do treat with DNase. So we want to get rid of, and RNA extraction still always has a little bit of contaminant DNA. So we treat with DNase to ensure that, and we use a very active DNase called turbo DNase, which is 1,000 times more active than for degrading DNA molecules. So we really want to get rid of all of the host, the human host DNA. With RNA, you can also, what remains is a large fraction of what remains is actually human ribosomal RNA. And so you can also do human ribosomal RNA depletion. And by doing so, you get a pretty good enrichment for both human RNA transcripts, which can be used for host response, as well as reads from RNA viruses or RNA from pathogens. So RNA is pretty straightforward. DNA is very challenging. And so the reason DNA is very challenging is that, especially as some of the speaker, some of the last two speakers have described, it's very challenging because for two reasons. One is that when you work with DNA, there's a lot of interest in doing metagenomic sequencing of tissues. Tissues like lung biopsy or liver biopsy or brain biopsy tissue have a huge amount of human host background. So that's very challenging. The second challenging aspect of DNA is if you want to look at non-sterile sites, and perhaps the other speakers can comment on that, but looking at, say, respiratory fluids, even BAL is less sterile than, because it has a microbiome, it's less sterile than, say, what spinal fluid would be. And so it's very challenging for those two reasons. And so for many groups, they have decided to try to do enrichment methods. There are advantages and disadvantages to different enrichment methods. Probably the most common enrichment method is done by Dr. Robin Patel's group at the Mayo Clinic. Her choice is to actually enrich specifically for, she uses a selective degradation method to enrich specifically for, say, bacteria and fungi by enriching for the cell walls. The problem with that, though, is that there's a cost to doing so. You can enrich for bacteria and fungi. You lose sensitivity when we did the testing for parasites, eukaryotic pathogens, and viruses. So if you really want to maximize your sensitivity, probably from body fluids, we've chosen to do a cell-free technique where we essentially remove the cells up front because there's too much background. And we're looking for essentially the signature in the cell-free compartment of say, a body fluid. If you have a tissue, though, as I said, it's very, very challenging. And we've technically resorted to simply brute force, which is essentially you just sequence more, just do deeper sequencing. Sequencing is becoming cheaper and cheaper over time. When we started, we were generating perhaps 1 million reads. Now we went to 10 million, 50 million. And I can tell you for, say, brain biopsy samples, we don't have to do a lot of them, but we sequence routinely more than 100 million reads in order to get the sensitivity that you need. But this is a very challenging thing in the field, how to basically enrich so that you can maximize your sensitivity. I'd be very interested in hearing from the other two speakers because they have more experience perhaps with tissue samples. Thank you.
Okay, thank you. 上海的胡碧杰教授，那刚才的问题您有什么评论吗？就是有关于简体的处理资料的一个去污染的这样的一个处理，在大陆您的经验如何？
就是这个是很关键的这个是问题啊。目前这个技术在临床应用当中是很有价值，但是也有一些不足的地方。刚刚我讲的稳定性不够啊，有的时候这个是核酸能监测得到，有的时候没监测得到，有的时候同样一份标本，一个高了，一个低了，那么就是有怎么样 来保持这个是良好的稳定性、重复性，这个是很重要。那么这是刚刚2位教授演讲的，我觉得确实这一个是，比如讲刚刚这个是支气管肺泡惯系啊 BAL， 这个是我们还是有一个是口腔的这个是污染菌，那么这种的污染菌怎么样来去除啊？这个是尤其是这个人源的这个是病原体，我们也有看到有一些农业的病灶，它这个是微生物，就想到的这个是农业嘛，微生物应该会多一点，结果反而阴性了。那么因为它这个白细胞这个是 DNA 比较多，反而就是我们把微生物的 DNA 覆盖掉了，这是不容易被检测出来了。所以这个是也是一个很大的这个是困惑的难题。所以这个是今后应该说从多方面着手吧。一个是怎么样这个是检出的这个是病原体的微生物尽可能高一点，包括我们讲分支杆菌，这分支杆菌的这个是我们就讲这个是结合或者 NTM、TB-NTM 这2个我们核酸序列就是提不高，然后通过有没有更好的方法，这个是让细胞不能破碎，让 DNA 能出来啊，这个是提升它的 DNA 的量。第二个呢就是人源的这个是标本怎么来一个是去除，这是2方面的走法。当然还有一个是我们能识别这个是人源的 DNA， 这个是也是一个是技术的走向。但是目前因为我是临床医生，更多的是一个解读临床这结果的解读，看看这个是结果对我这个是临床有多少大的一个是支持帮助。那么我能从临床的角度讲起来啊，这个是无菌部位啊，这是由正常菌群的污染的部位啊，这个临床过程符合不符合这个病原体引起的，也有的是非常奇怪的状况，可能是全国第1例报告，甚至全球第1例的报告之前没有报告过，也有可能是根本就是一个环境污染菌。那所以你这个是罕见的疾病，跟我们的这个是环境污染菌怎么来鉴别？所以我觉得应该是这个技术是方向是好的，但是怎么样去完善是下面一段时间我们的这个是科学家，这个是技术人员需要做的事情了啊。我临床医生可能更多的是这正确的应用和解读的问题。
Yeah, so interpretation by a clinician is very important after the result was noted. Dr. Liu.
Thank you。其实我可以跟大家分享的是失败的经验啊，就是因为之前就是为了要想办法降低那个 host 的一个污染，我们做过很多，现在看起来可能大家觉得不尝试，我们试过把血球降下来，用各种不同的离心梯度去做这件事情，然后我们也试过用，因为人的 DNA 是 linear。所以我们也试过用 linear DNase 去处理这个事情，甚至用各种不同的一个过滤的方式，想办法把 host 的一个污染或者是其他污染降到最低。大概都尝试过，不过我想其实就像是 Dr. Chu 讲的，其实都是 trade off， 就是不管任何一种方法，它都得付出代价，就是没有一个方法说就是有一好没两好的概念了。我想这是我们的一点心得是。
Thank you for your comment from Dr. Liu. Professor Huang.
I have 2 questions, one for Dr. Hubije and one for Dr. Wilson. Can I ask the first question first with Mandarin? 所以胡教授，那个你的 NGS 的经验是非常的 impressive 了哈。那你可不可以告诉我们大陆目前应用到什么地步了？然后有什么障碍让这个技术没有办法大量的使用？是钱的问题呢，还是给付的问题，还是保险的问题，还是什么？
好，谢谢你，这个是很好的问题。大陆目前这个是越来越多的这个是临床医生开始使用的这个是2点测序，但是呢，这个是叫mNGS 呢，在医疗机构很多的医院是不能收费的，所以我们更多的是第三方检验啊，第三方检验呢，就是相对讲起可及性啊、方便性啊，这是在大城市、中型城市可以。但是呢，一些小城市呢，可能要物流、要快速运输，相对稍微这个是难一点。但是不管怎么样呢，因为越来越多的临床医生像我一样，像我2017年的时候尝到了甜头以后，这个是之前的 疑难的感染病原体一直搞不清楚，用了很多的抗生素无效的情况下面，用了这个东西蛮好的，所以就是会临床平时碰到的有一点复杂疑难的都会想到这个技术。所以目前应该说至少我们这个是3级医院吧，这个是4级医院、省4级医院，这个很多的临床医生，尤其是 ICU 的医生、呼吸科医生、感染病科医生、血液科医生啊，基本上就是相对比较常规的啊。这个是也有一些骨科的医生，他就碰到一些骨科的病灶处理不了，是个感染，那么这个是非常越来越广泛的使用。那么有一点呢，我说可能之前我跟美国的华人专家是深层次的交流过，他说这个是为什么中国大陆二代基因测序这个是用了这么好，甚至比美国用的好。我现在不敢说这个美国用的怎么样，刚刚这个是美国科研做的很深入啊，但是临床的到底怎么样？这个邱教授等会再可以解释一下。但是呢，我们的临床应用是真的是非常多。其中一个非常大的原因是什么？中国大陆的常规的微生物 CS 的检测水平比较低，很多的它这个是微生物检测不出来，甚至我们讲这个一些之前我们讲稍微少见一点复杂的病原体，这个是我们讲流感嗜血杆菌，对一个基层医疗机构它就不容易检测得到，但大医院是没有问题。那在大医院有个问题是叫 NTM。还有是 nocardia 这种的病原体，还有复杂一点的这个是方甲，这个是它就检测不出来，所以导致了我们对这个是分子诊断技术啊，我不要动脑筋，我只要有钱啊，这是我们现在是3000多块钱，相当于是，这是如果美金比1比7点几的话，这个是500美金500美金啊，这个是做一个标本。但是有的时候真的是很难的时候，他有做，但是因为大陆的整个人群是比较多嘛，所以这个是我们用的会比较多。那么我中山医院呢，现在有的时候还随访的，就是第一次做了，第二次还继续随访。那么有很多的医院可能就不随访了。那这个是我的随访呢，很多是免费给他的，就是我第一次付费了，第二次不付费了。所以您刚刚讲的另外一桩事情，就是我们费用是问题吧？费用肯定是个问题。那么现在我觉得还是比较高，但是我们大陆的病人数量太多，所以导致了现在我们的绝对的数量，目前看上去这数量监测2代基因测序真的是蛮多的。我中山医院现在可以这样说，一天要做10到20个，这是2代基因测序做微生物的检验，所以这个应该还是我认为啊，这个还是有点多的谢谢。
I have a question for Dr. Wilson. In your slide, you said that the autoimmune encephalitis is increasing. The incidence seems to be increased. So could you tell me why? Is it the true increase, or is it because of the increased detection diagnosis of autoimmune encephalitis? And I'm a pediatrician, so in my daily practice, I've seen more and more use of past steroid in our patients. Whenever there's a kid with chronic symptoms, neurological symptoms, and if the microbiology diagnosis turns out to be negative, and it's very likely he will be put on a past steroid. So can you comment on the statement that if the mNGS reveal no pathogen in the CSF, and the kid have chronic symptoms, can we just put him on the past steroid? And is there any caveat or any problem with that?
So those are great questions. The second one is not easy. For the first question, I don't think that I don't know, but I don't think that the prevalence of autoimmune encephalitis is actually increasing. I think that the diagnostic testing just keeps getting better. I think we've all recognized there's been now a yearly or even more than once a year, new autoantibodies being identified with these conditions, and with increasing availability of these autoantibody panels that one can send, to look broadly for a variety of autoimmune causes of encephalitis. I think we're just detecting many cases that used to go undiagnosed, some of which got were blamed on viral infections. And then for the question of, so I'm not a pediatrician, but I think even when I am faced with using pulse steroids in adults, I think it's a very tricky question. I think and of course, you know, a negative sequencing result, I think we all believe that has some predictive ability to suggest that, you know, there's at least not an active infection, but it's definitely another step to then say it's an autoimmune disease. And I think unless there's a good clinical story that really seems convincing for an autoimmune condition, I get nervous by treating with steroids, especially if the condition is chronic, because, you know, it's hard to know what biomarker to follow, and is a way to judge success or failure of the treatment. And as everyone knows, you know, the long-term complications of steroids are significant. So it can become quite tricky. So there are certainly instances where it makes sense, especially if the patient is declining and you need to do something. But I think they're probably more often than not, there's reasons to hold off and just continue to try to get more diagnostic information.
Okay, thank you. There are some questions from the online participants. The first question is to Professor Chu. Could you have some comment or explain about the difference between your nano technique and the traditional Illumina test for NGS? What is the benefit for your nano technique versus Illumina traditional method?
Sure, I'd be happy to do so. So much of sequencing is done on worldwide is done on Illumina instruments and also on BGI instruments. These are typically benchtop sequencers. So they're sequencers, they're instruments that you would place in the laboratory, and they have the benefit that the yields are typically very high. You can generate, for instance, you know, anywhere from 30 million to more than 3 billion reads a run. So it allows you to do high throughput sequencing. So if you want to do metagenomics, say in a laboratory where and you want to run say metagenomic testing on a large number of patients, you know, I would recommend using one of the high throughput sequencers, and you know, by BGI or by Illumina or and there are a few other companies as well, like Iron Torrent. The Oxford nanopore sequencer, so this isn't my technique. It's actually a commercial sequencer, and it has the benefit that the MinION, which is the instrument that you saw in my presentation, it's a pocket-sized sequencer, so it's very small. You can fit it in your pocket. You can bring it in your backpack. You can put it in your, and we have actually been able to essentially be able to do sequencing in the field or sequencing while traveling, what we call a laboratory in the suitcase. The idea is that you can do sequencing and deploy it very widely. It also makes it easier if you have a sequencer, a nanopore sequencer, of implementing the sequencing in say a rural hospital, or perhaps in a laboratory, or say in the emergency department in what we call point of care settings. So if you're interested in doing that, that's the benefit. I believe that's one of the major advantages of nanopore sequencing. The other difference is that Illumina sequencing is sequencing by synthesis, meaning that you have to sequence everything. The yield is very high, but you have to wait until the run is done. And the run typically can take anywhere from 17 hours to 24 hours. So because of and that's just the time to do the sequencing. So because of the turnaround time, it's impractical to be able to get, say, a result. Say it usually takes at least 24 hours. So you're going to get a result the next day at the earliest. And the advantage of a nanopore sequencer is that you can start sequencing and analyze the data as soon as you start sequencing. So as I showed you in the example, you would be able to get a result very quickly and actually be able to get a same day answer. And so it really depends on the disease that you're looking at, the infection. If you have a critically ill patient in the hospital with, let's say, a sepsis, unknown sepsis, where you need to make the diagnosis very quickly, well, then nanopore sequencing will be more attractive. If you have lots of patients that you need to sequence, or if you have, neurological infections, for instance, where we talked about having low titers, and if you absolutely want to use the most sensitive method of sequencing, then one of the higher throughput sequencers might be a better answer. As in anything, there's a trade-off. There's no, I believe there's really no one best method. You have to look at your application and to be able to see what sequencer or what approach would be most clinically useful.
Okay, thank you. The second question is for Dr. Wilson. Is it possible to integrate the transcriptome test and the pathogen detection into one test?
So I think absolutely that's the kind of evolving story there, is that the data all come from the same experiment. There's no additional laboratory work that's required to generate both sets of data. Right now with the clinical test at UCSF, the validated part is the pathogen detection from spinal fluid and plasma, but then on a research basis, the human RNA sequences can be analyzed to look for the spinal fluid test, can be analyzed to look at the host transcriptome. That part hasn't been clinically validated yet, but the analysis is available on a research basis. So that's the dream, is to have both clinically validated and to be able to provide a combined analysis.
Okay, thank you. The third question is from National Taiwan University Hospital, Dr. Chen Baoyu. He would like to ask Professor Chu about the sensitivity diagnosis of NGS in fungi infection, because Dr. Chen Baoyu had some experience that is difficult to diagnosis of fungal infection by NGS study compared with the traditional method. So how is your comment about the diagnosis sensitivity of NGS in fungi?
No, it's a very important point. I would say that fungi and mycobacterial infections are very difficult to diagnose primarily because of that. And we do know that in the setting of documented fungal infection, that the titers are lower than you would see in, say, a bacterial infection, for instance, or most bacterial infections. And so the answer to your question is about sensitivity is, I would say it really depends. One question I would ask is, like, what sample type are you looking at? Because one of the key limitations of metagenomic sequencing is that we do metagenomic sequencing from one sample at a time. And the issue with fungal infection in general, you really want to be able to look at tissue. Because that's where the fungus, especially an invasive fungal infection, that's kind of the site of where the infection is located. So the sensitivity, the analytic sensitivity, if we're able to report, say, subthreshold results, we found that the analytic sensitivity is pretty good. I mean, it's down to, say, it's essentially equivalent to PCR, down to, say, 1 to 10 copies per, say, mil of fluid. Now, the problem, though, is that that doesn't mean, although the sensitivity could be close to, say, could be more than 95%. The problem is that that doesn't mean that the clinical sensitivity is more than 95%. The clinical sensitivity still ends up being lower, and that's because we're looking at the fluid. But again, if there are no sequences, or there's no DNA from the fungus in the fluid at the time that you obtain the fluid, you're not going to be able to diagnose the infection that way. So it's either, I feel that to maximize sensitivity, we either have to move to tissue, start looking at exactly where the site of the pathogenesis is, or the kind of adopt, say, a host response method, couple the fungal detection with, say, a host response analysis, which we have found maybe was very promising with respect to distinguishing between, say, bacterial and fungal infections.
Okay, thank you. The last question is about one doctor say in his hospital, he had a multiplex PCR test. How is your opinion about the clinical application of multiplex PCR versus NGS in pathogen detection? Can Professor Chu or Dr. Hu Bijie, the multiplex PCR and the versus to NGS in diagnosis in infectious disease? How is the difference in your opinion?
Yeah, Dr. Hu Bijie, do you want to comment? I have a clear opinion on this, but I'd be interested in hearing from you.
要是您再补充，这个是确实两个技术都是分子诊断技术，然后呢，我们现在这个是感染病科呢，都两方面都在使用，但是总体上讲，就是我们的2对基因测序的整个一个是常规的流程时间还比较长。PCR 呢，这个时间是明显要缩短。第二个，这个是我们的 PCR 的费用要少得多，精准性呢，准确性要强得多，这个是敏感性啊，这个讲的。但是 PCR 的最大的这个是缺陷跟2对测序呢 ，PCR 是我要知道这个病原体是哪一些病原体，然后我才可以去做。如果我不知道的病原体的情况下面，那就是2对测序可能这个是更有优势。但是如果我比如现在，比方我刚刚讲的，我好多的胸部 CT 的肺部感染，慢性肺部感染有相当部分我都知道，哎，这个病人应该是说我只要明确他是这个 NTM 的是哪一种，那我们可以用一个 kit， 这是 NTM 的多重 PCR， 它有这个是常见的临床有20几种啊或者30几种，这个是有些有商品化的这个 kit 可以用，然后我这个是直接可以告诉你，那么这个是价钱便宜，这个是敏感性高，然后速度快。但是呢，我不知道情况下，你以为是这个 NTM， 其实是它有可能是卢卡氏菌，或者是另外一个病原体，那你就漏检了。所以我不知道的情况下面，一网打尽的话，那些这个是2对基因测序。但是我只要说我就这么几点病原体，那我就是可以用多重 PCR。那么在大陆呢，现在这个是也有一部分的公司一边是在做那个2对基因测序。还有一个呢，就是在做这个是多重 PCR， 这是目的就是说两方面需要不同层次的病人的需要，不同场景的病人的需要。如果他这个是要快速，就是我常见的，比方中枢感染就那么几个病原体，或者是我败血症我可能就那么几个病原体，或者尿路感染就那么几个病原体，那我可以按照常规的去测试，这样这个是便宜快速，这个是敏感性高。但是我们搞不清楚的时候，尤其是常规一圈打了失败了，还是病原体没找到的话，那就还是有2对测序更有这个是它的一个优势啊。不知道对不对啊？这个邱教授，请您这个是。
No. I completely agree with Dr. Hu in that it's right now. I mean, the key advantages are exactly as he said, that PCR is faster, it's cheaper, but the key disadvantage is you have to know what you're looking for. I mean, if what you're looking for is not on the panel, you're not going to be able to detect with multiplex PCR. However, I don't feel that metagenomic sequencing is going to replace multiplex PCR because of that reason. I mean, I think we all have the vision or perhaps the wish that eventually sequencing will become cheap enough, fast enough that we could simply replace it. And I do think that it has the potential, as sequencing becomes cheaper and cheaper, we will be able to, run this on more and more patients and be able to do it at far lower costs and do it with more rapid turnaround times. I think if we do get there, then I think eventually metagenomic sequencing has the ability to potentially replace all direct detection methods. We still will probably need serology because that's an indirect detection method, or host response analysis. I mean, those are indirect ways of diagnosing the disease. But with respect to culture, PCR, antigen testing, I mean, potentially sequencing may replace it, but we're not there yet. It's still too expensive, and we're still kind of exploring. So I actually feel that it's still an emerging technology, but you know, it's my hope that we can democratize it so that it would be available really widely to every patient, because I feel that, you know, patients overall will be able to benefit from having this test available.
Okay, thank you. Maybe two steps of diagnosis emergency department is required at present. One is for the traditional method for detection of pathogen, including the multiplex PCR. If the diagnosis remain unknown or uncertain, then the NGS could be the next step. So because of the time limited, I would like to move to the last section, Professor Wang for his ending.
谢谢盛教授哈，那谢谢今天5位的主持人哈，陈义军陈教授、黄立敏黄教授、邱南昌邱教授、李明士李教授哈，还有沈婉辉盛教授的主持哈。Thanks for speakers Professor Chiu, Professor Wilson 好，那还有就是胡碧杰胡教授哦，那刘博宇刘主任的精彩的演讲 ，for your excellent talk。那今天非常感谢哈台中荣民总医院，还有台湾医师检验协会，还有亚洲准医共同举办了这一次的研讨会了哈。那台湾这个，我想近20年来，全球都是出现了很多的传染病的流行哈，那大概都是来自于新型的来源哈。那像现在流行的这个，现在正在进行的这新冠病毒哈，那频繁的群突发之下，次世代的定序的技术的病原基因比对的优势哈，能够提供全新的应对的方法哈。哦，那在会议中呢，Professor Chiu也分享了技术应用在诊断 SARS-CoV 的案例哈，那也成为准确的及鉴定疾病的来源、时间、传播跟扩散哈。那是一个不可或缺的工具了哈。那目前临床诊断技术的局限哈，在造成很多的脑炎啊、菌血症、肺炎等感染疾病哦，这个多样性的可能的致病源哦，难以诊断判断的原因哦啊，这些病人可能也会造成快速的恶化哦。那需要更及时、更敏感的诊断工具来制定后续的临床决策哈。那Professor Wilson 也借由定序技术来找出脑炎跟病原基因的关联，那有开拓了新的诊断跟治疗的一个方向啊。那定序技术迅速的发展那逐渐成熟的大环境下，使得我们可以将先进的定序的科技应用在临床的感染症上许多国际间的尖端研究正在广泛的使用这种次世代的定序来侦测许多多元的病原体那今天胡教授也分享了在上海在中国可能是做这种次世代定序可能是最多的国家医院等等那刘主任也分享了在台湾的一些临床的经验那透过这个研究实例的研究次世代定序这些没有培养不需要假设病原体的检验技术能够诊断出检体内的微生物的比例跟基因资讯，那开拓临床感染应用的无限可能那也指出未来临床诊断趋势可能走向一个数据导向的一个决策的过程那最后这个因为现在都在推广这个精准医疗精准医疗的普及将有更多新颖的研究跟技术来运用在临床感染症上那常规检验的方法可能有时候没有办法诊断出这个重症的感染病患 那透过次世代的定序技术，能够解决现有的检测的局限那辅助临床医师综合判断的治疗那借由这一次也跟3位国外的演讲者共同的学术的交流那能够持续的再继续的我们学习跟反馈那期盼在不久的将来这个次世代的定序的技术能够导入在台湾的临床研究上面临床的诊断上面，那提供大家多元的武力来对抗这个感染的疾病那谢谢大家今天的参与谢谢大家那希望我们未来还有机会，就是说大家再来多分享慢慢的这个次世代的这个检验的技术能够在台湾能够继续去推广那让这个精准医疗我们现在的精准医疗能够更能够诊断出一些病原剂能够让病人得到更加的一个治疗以上好，谢谢大家。